Prokaryotes. Imagine living all your life as a member of what you believe is the only family on your street. Then one morning you open the front door and discover houses and neighbors all around you. Where did all the people come from? What if the answer had turned out to be that they had already been there? You just hadn't seen them. How would you view the world now? When the microscope was first invented, we humans had just such a shock. Far from being alone, we share every corner of our world with microorganisms. Even a seemingly clean toothbrush contains a film of bacteria on its bristles. How are prokaryotes classified? Prokaryotes are classified as bacteria and archaea, two of the three domains of life. The smallest and most abundant microorganisms on Earth are prokaryotes, unicellular organisms that lack a nucleus. Prokaryotes have DNA like other cells, but their DNA is not found in a membrane-bound nuclear envelope as it is in eukaryotes. Prokaryotic DNA is located in the cytoplasm. A bacterium such as E. coli has the basic structure typical of most prokaryotes: peptidoglycan cell wall, an outer wall, and then cell membrane. And inside that is the DNA with the ribosomes and flagellum. Recently, biologists have divided prokaryotes into two very distinct groups, bacteria and archaea. These groups are very different from each other. Therefore, biologists now consider each group of prokaryotes as separate domains. Eukaryotes are the third domain. The larger of the two domains of prokaryotes is bacteria. Bacteria include a wide variety of organisms with lifestyles so different that biologists do not yet agree exactly how many phyla are needed to classify this group. Bacteria live almost everywhere, in fresh water and salt water, on land, and on and within the bodies of humans and other eukaryotes. Escherichia coli, E. coli, a typical bacterium that lives in human intestines, is shown. Bacteria is usually surrounded by a cell wall that protects the cell from injury and determines its shape. The cell wall of bacteria contains peptidoglycan, a polymer of sugars and amino acids that surround the cell membrane. Some bacteria, such as E. coli, have a second membrane outside the peptidoglycan wall that makes the cell especially resistant to damage. In addition, some prokaryotes have flagella that they use for movement, or pili, which in E. coli serve mainly to anchor the bacterium to a surface or to other bacteria. Under a microscope, archaea look very similar to bacteria. Both are equally small, lack nuclei, and have cell walls, but there are important differences. The walls of archaea lack peptidoglycan, and their membranes contain different lipids. The DNA sequence of archaea genes are more like those of eukaryotes than those of bacteria. Based on these observations, scientists have concluded that archaea and eukaryotes are more similar to each other than to bacteria. Chromosomal DNA, plasmid DNA, ribosomes, cytoplasm, plasma membrane, cell wall, and the capsule. And here are flagella. Many archaea live in extremely harsh environments. One group of archaea produce methane gas and live in environments with little or no oxygen, such as thick mud and the digestive tracts of animals. Other archaea live in extremely salty environments, such as Utah's Great Salt Lake, or in hot springs where temperatures approach the boiling point of water. Prokaryotes vary in their size and shape, and in the way they move, and in the way they obtain and release energy. Prokaryotes range in size from 1 to 5 micrometers, making them much smaller than most eukaryotic cells. Prokaryotes come in a variety of shapes. Rod-shaped prokaryotes are called bacilli. Spherical prokaryotes are called cocci. Spiral and corkscrew-shaped prokaryotes are called spirilla. Prokaryotes can also be distinguished by whether they move and how they move. Some prokaryotes do not move at all. Others are propelled by flagella. Some glide slowly along in layers of slime-like material they secrete. Prokaryotes need a supply of chemical energy, which they store in the form of fuel molecules such as sugars. Energy is released from these fuel molecules during cellular respiration, fermentation, or both. Prokaryotes vary in the ways they obtain energy and in the ways they release it. Notice that some species are able to change their method of energy capture or release depending on the conditions of their environment. Heterotroph, other feeders. Photoheterotroph, light and other feeders. Photoautotroph, light self-feeder. And chemoautotroph, a chemical self-feeder. And for nutrition and metabolism, obligate aerobe, it requires oxygen, and obligate anaerobe requires a lack of oxygen. Facultative anaerobe, surviving without oxygen when necessary. When a prokaryote has grown so that it nearly doubles in size, it replicates its DNA and divides in half, producing two identical cells. This type of reproduction is called binary fission. Because binary fission does not involve the exchange or recombination of genetic information, it is an asexual form of reproduction. When conditions are favorable, prokaryotes can grow and divide at astonishing rates, some as often as every 20 minutes. And this is a time lapse. When growth conditions become unfavorable, many prokaryotic cells form an endospore, a thick internal wall that encloses the DNA and a portion of the cytoplasm. Endospores can remain dormant for months or even years. The ability to form endospores makes it possible for some prokaryotes to survive very harsh conditions. The bacterium Bacillus anthracis, which causes the disease anthrax, is one such bacterium. These are the green endospores inside the bacterial cells. And here's the endospore inside the bacterial cells. Many prokaryotes exchange genetic information by a process called conjugation. During conjugation, a hollow bridge forms between two bacterial cells, and genetic material, usually in the form of plasmid, moves from one cell to the other. Many plasmids carry genes that enable bacteria to survive in new environments and to resist antibiotics that might otherwise prove fatal. This transfer of genetic information increases genetic diversity in populations of prokaryotes. What roles do prokaryotes play in the living world? Prokaryotes are essential in maintaining important aspects of ecological balance in the living world. In addition, some species have specific uses in human industry. Bacteria called actinomycetes are present in soil and in rotting plant material, such as fallen logs, where they decompose complex organic molecules into smaller molecules. By decomposing dead animals, prokaryotes supply raw material and thus help to maintain equilibrium in the environment. Bacterial decomposers are also essential to the industrial sewage treatment, helping to produce purified water and chemicals that can be used as fertilizers. Cyanobacteria in the genus Anabina form filamentous chains in ponds and other aquatic environments where they perform photosynthesis. Photosynthetic prokaryotes are among the most important producers in the planet. Food chains everywhere are dependent upon prokaryotes as producers of food and biomass. All organisms need nitrogen to make proteins and other molecules. Nitrogen gas, N2, makes up 80% of the Earth's atmosphere, but only a few kinds of organisms all of them prokaryotes, can convert N2 gas, nitrogen gas, into useful forms. The process of nitrogen fixation converts nitrogen gas into ammonia, NH3. Ammonia can then be converted to nitrates that plants use or attached to amino acids that all organisms use. Nitrogen fixing bacteria and archaea provide 90% of the nitrogen used by other organisms. Some plants have symbiotic relationship with nitrogen fixing prokaryotes. The bacterium Rhizobium grows symbiotically in nodules or knobs on the roots of legume plants such as soybean or clover. The Rhizobium bacteria within these nodules convert atmospheric nitrogen in the air into nitrogen compounds essential for plant growth. Prokaryotes, especially bacteria, are used in the production of a wide variety of foods and other commercial products. Yogurt is produced by the bacterium Lactobacillus. Some bacteria can digest petroleum and remove human made waste products and toxins, at least toxins to us, from water. Other bacteria are used to synthesize drugs and chemicals through the techniques of genetic engineering. Bacteria and archaea adapted to extreme environments may be a rich source of heat stable enzymes that can be used in medicine, food production, and industrial chemistry.